So welcome everybody. The idea of having this afternoon of talks is to illustrate some ideas, give some examples, I should say, of where bringing these very different communities together during the last 6 months has resulted in new research and new directions, as well as bringing people who don't normally work together to work on different things. And so we'll have a number of talks today, and then a sort of discussion at the end. So the first speaker then is Duncan, and he's going to be talking about modeling, collective movement, which was the core of the whole meeting. Okay, so thanks, Duncan.
Yeah. Thank you, Philip, and thanks for the rest of the organizers of the Mathematics of Movement program for letting me talk to you today about some of my experiences as a Newton-Simons postdoctoral fellow here. So just to give you a little background on my research, I come from more of the mathematical modeling of collective cell migration field. And a lot of the themes of my research in my PhD and my first postdoc have been looking at mathematical modeling of cell groups in order to gain some experimentally relevant insights. So the photos and videos that I'm showing on the right here depict neural crest stem cell migration in developing chicken embryos. And during the course of migration, these cells, which are highlighted in green on the top here, form these nice, robust, anisotropic ribbon-like streams as they travel to some kind of target site in the embryo. And depending on the final location where these cells end up, they become skin cells, bone cells, cartilage cells, what have you. And a key question we wanted to address with a mathematical model was just how do cells further behind this kind of front of cells, how do they sense their leaders, and why don't they move off into these neural crest cell free regions that are bordering those streams? And so in collaboration with some biological collaborators in the United States, we had started looking at the ability of cells to remodel their environment, and specifically extracellular matrix proteins known as fibronectin, and looking at how that confers nice robustness to migration by having this kind of positive reinforcement mechanism informing cells behind where the cells in front of them have gone. And we used that mathematical model to make predictions as to experiments that our collaborators had been proposing, and used it to kind of further guide their experimental design for the future. The other broad theme of my research has been looking at the relationship between ways in which you can mathematically represent collectives from individual-based models where you actually track each cell. So for instance, this is a model looking at the formation of blood vasculature, where you have migrating cells highlighted in blue there, traveling towards the right-hand boundary. As they move, there's red cells that divide along their path, and that's what's creating this new kind of vascular structure that would eventually oxygenate, hypoxic tissues. But as the number of agents or individuals in your model increases, these become a lot more intractable to simulate computationally, and become harder to analyze as well. And so what I've had spent some of my research doing was just looking at mean field approximations, so continuous models of these kind of individual-based descriptions, and trying to compare them. So the idea is, well, we can't get information on individual branching structures because you're looking at a kind of ensemble average result. You can still obtain some experimentally relevant insights, such as how fast these cells are traveling and what's the approximate density that you'd expect to find. And so you can show that we're trying to find biologically relevant scenarios for which these continuous models match well with the experimental or the individual-based results that you'd hope to find. So that kind of leads to where I essentially started here at the Mathematics of Movement workshop. I was hoping to kind of extend and branch out into a couple of different questions in this field, one of which being this idea of model calibration. So how you can actually connect these mathematical models to the high resolution spatial and temporal data that we can collect now. So up at the top left, up at the top, that's a video of neural crest cell migration. It can actually track the individual movement of cells, and how can we relate that to the mathematical modeling that we do, as well as taking a look at the roles of individual heterogeneity and the environment on cell migration. So how does the ability of individuals to specialize in, say, like a migratory phenotype or a more proliferative phenotype affect the behavior of their groups? And then how do they encode that behavior in their environment, and vice versa? How does the environment dictate what the group eventually does? So you can imagine there's a tumor spheroid experiments on the bottom here, showing that if you have different cells cell with different phenotypes placed in that tumor. You can have either a spheroid morphology, a branch morphology, or some more kind of elongated structure. So the rest of my talk is essentially just going to be a whistle-stop tour of different projects that I began here at the Newton Institute that are hoping to address some, if not all, of these questions. They're in a variety of different fields, including zebrafish patterning, blood vessel development, and the third one is lion migration. So in terms of the zebrafish patterning, we're working with, say, Jose Carrillo, Alexandra Volkening, Marcus Schmidtchen, Shakar, and Josh Bull on trying to understand how different striping patterns arise in zebrafish. So basically, how do zebrafish get their stripes? And Alexandria, pictured here, had come up with an individual-based model that essentially was able to describe the stripe formation in terms of the interactions between two different cell types, these black melanophore cells and these yellowish xanthophore cells. And this model includes the effects of migration and birth and death of these cells. But the problem is the cell numbers are too numerous for her to efficiently simulate with this agent-based model. It just simply takes too long to gain some experimentally relevant insights. And so the idea that we had was, well, why don't we try to construct a continuous version of this model? We try to replicate some of the rules that she has at the individual-based scale. And because it's easier typically to analyze a PD model than a stochastic model, we might be able to get some of the questions that she wants to address, such as what are the mechanisms that determine the stability of your patterns? The other thing that we're interested in is how do you actually quantify and robustly characterize the kinds of patterns you can get from these types of models. So I showed you this example of striping patterns, which you see in wild-type embryos, but in mutant embryos, you can get more like spotted patterns or even labyrinthine patterns like you see at the bottom here. And how can you actually quantify those different patterns? Because you want to, well, what we're aiming to do is start off from a completely random initial condition and have the patterns arise from there. But you can imagine that if you take the ensemble average of all of these things, you're just going to get a completely uniform, approximately, solution. But nevertheless, you still get similar kinds of patterns, each different realization. Like they might have the same length scale or the same number of spots in your image. So how can we actually quantify all that? So in terms of just matching these continuous and individual-based models, we decided to work with a modular approach here, where you start with your individual-based model for migration only, which is essentially just a Newton's overdamped version of Newton's law with a conservative potential representing cell attraction and repulsion. You can see an example simulation in this video on the bottom here with the little black cells. The colors just represent the ensemble average of 10,000 simulations of this agent-based model. So basically, how many cells do you expect to find in this little location on average? And you can look at the weak formulation of this ODE, take the limit as the number of cells goes off to infinity, and you can actually derive an integral differential equation, so a PD version of this model, in which you're tracking how does the number density of cells evolve over time and space. But again, because you're only dealing with a finite number of cells, in this case, 400 to 1,000, rather than say like billions of cells, we've added in this scaling parameter alpha here, just as a way of just correcting for these finite number discrepancies that you'd see. And what we've been doing is just fitting this value of alpha here to ensemble average agent-based model results. The solution of that PDE is shown in the video on the bottom in terms of the colors. And then we just plotted on another agent-based model realization just to verify that you can actually fit the PD quite well. And again, you can compare this for different types of initial data from, say, like initial condition where you have a box or a stripe or two stripes or whatever. But in all cases, you basically find that if you fit the ensemble average results, you get the same value of alpha no matter what you find. So it's giving us a nice support for the idea that you have this inherent timescale at which these ensemble average solutions are evolving at, and it's different from what you'd expect from the mean field scaling. The mean field scaling you expect to just be equal to 1. And we can take this idea for the migration model, do it for a model for cell birth and death, and combine the two together, and you can find that you can have a really good match in terms of not only how many cells you end up with over time, but also how far away these things spread out. If you're just looking at, say, the support of the PD versus the ensemble average individual-based result. Right. And then in terms of like actually quantifying these patterns, what we've been doing is taking our individual based model, creating example striped patterns, spotted patterns, labyrinthine patterns, and applying different types of statistics that can actually extract some kind of spatial information. So the first one we're taking from inspiration from Joshua Bolt, who's out in Oxford. He's been looking at spatial statistics known as pair correlation functions, which essentially just mean you take for every single cell, you start looking in the little annular region around your body. You count how many cells are found within that radius of some outer radius R. Average over all the cells in your simulation, and then further normalize it by the number of cells you'd expect to find within that annular radius if you just had completely spatially uniform distribution. That's what this equation here on the top right is just supposed to represent. But the output that you get of this kind of pair correlation function is these kind of oscillatory waves, where if the way to interpret this graph, if you're at a value equal to 1, that's a clustering you expect to find with complete spatially uniform randomness. If you're a value above 1, you have more clustering than you'd expect to find from a uniform distribution. If you're below 1, less clustering. So essentially, the peaks of these waves tell you where you expect to find more cells. Troughs tell you regions of like empty space. But what's more important here is kind of the frequencies that you would get from these graphs, or the wavelength you get from these graphs, because they tell you information about how far cells are away from each other. So cell-cell distances and longer wavelengths would correspond to more longer scale patterns, like the striping pattern you see there. We've also been looking at other types of statistics, such as those inspired from biology. So the biologists have a way of trying to characterize spots versus striping patterns. And it's essentially identifying a connected component in an image, trying to calculate the area and perimeter of that connected component, and essentially just computing the ratio of area to perimeter squared. So effectively, they call this a pattern simplicity score. A value of 1 is going to correspond to a circle. A value of 0 is going to correspond to an elongated structure, like a very long stripe, or like a labyrinthine structure. And so what we're trying to do at the moment is just collecting all these different metrics on different realizations of these patterns, seeing if they're actually robust to the just the initial random seed or stochastic simulation. And what we want to be able to do is combine these different statistics into a signal signature that would distinguish between a striped pattern versus a labyrinthine pattern versus a spotted pattern, and use those signatures to actually fit our PDE model to, using, say, like approximate Bayesian computation. And once we have this kind of description, we can use that to create an accurate continuous model, which we can then analyze with linear stability analysis, all the typical tricks you would do. All right, so that's the zebrafish project, but now I want to switch gears and look at now the realm of angiogenesis. And this is in collaboration with Helen Byrne, Daria Stepanova, Tomas Alarcon, Yi Zhang, and Simon Siga. And again, we're looking at angiogenesis, the formation of blood vessels. You can imagine this forms very nice branching networks, such as the one seen at the top left here. And just for the purposes of the presentation, all you really need to know is that this process is caused when hypoxic tissues release this chemical known as VEGF into the environment. VEGF diffuses to the blood vessels, and then when it reaches the blood vessels, it activates cells lining the blood vessel wall to adopt one of two phenotypes, one being a migratory tip cell phenotype, which travels towards the source of VEGF, the other being a more proliferative stalk cell phenotype, which essentially is dividing along the path of these cells. And there's been a variety of different mathematical approaches that have been done to try to model this angiogenic process, from continuous-based models down here to more discrete ones over here. But what we're interested in right now is the effect of the ability of cells to change phenotype. So just because you're a tip cell at the beginning of an angiogenesis realization doesn't mean that you remain a tip cell experimentally. There's evidence that you actually can adopt a stalk cell phenotype, and there's some cell overtaking. But the question that we want to address is, what's the role of this ability to switch phenotype at the types of branching networks that you can form? So rather than looking at branching at the head of these angiogenic fronts, we want to look at how does the ability to switch from a stalk cell phenotype to a tip cell one cause you to form branches far behind where these cells are migrating. and how does that affect the kinds of statistics and networks that you would form. And the other one we want to look at, and this is just with Helen Byrne alone, is trying to identify like a minimal model that has cells overtaking one another, and if we can actually accomplish that in a pretty simplistic fashion. So the idea that we have is to take a model for cells switching phenotypes, which is from Daria. So it's a model taken from her original paper, which allows cells to switch state stochastically between a tip cell state and a stalk cell state, depending on how much VEGF you sense in a neighborhood around yourself. We want to combine that with a model known as a lattice gas cellular automaton in order to form larger scale branching networks than Daria can already do. And once we are able to generate these large scale networks on a very fast time scale, we're able to now generate these networks, but also compute some metrics relevant for branching, such as how many tip cells you have in your simulation, but also some more topological statistics, such as those you would get from persistent homology and topological data analysis. So essentially looking at the number of connected networks, the number of holes in your networks and stuff like that. And that one we can rely on existing software from John Nardini, who's already looked at agent-based models of simpler agent-based models of angiogenesis and has that kind of that toolbox already set up. And in terms of the cell shuffling problem, so this video here just shows an example where you have tip cells at the front that are eventually overtaken by these yellow cells behind them. And one way in which this could occur is just through cells just encountering some highly heterogeneous environment. And because of this, having to overcome the effect of this environment, they have to push through, they have to deplete their resources, sort of like proteins or something like that. And because of that, they might like lose the ability to maintain their position at the front and then eventually just get pushed aside by their neighbors. And you can see like a similar thing is kind of happening here in neural crest cell migration, where you have green cells at the start, which are eventually overtaken by these purple cells. And so we want to just see, like, can we identify a minimal model where you can actually get this overtaking to actually occur, where one cell from the back reaches the front. And so this is still like a work in progress. We're still trying to come up with this kind of model. But the idea is that you would have cells moving through some kind of viscous environment. So you'd have a kind of drag force. Cells can also repel and attract each other, just as before. But you'd add in this additional like active propulsion force, which depends on some kind of internal cell state represented by this U variable here. And you would have some kind of internal dynamics from this, but also some additional term representing the influence of the environment. So what I'm trying to show here, these yellow cells are the cells moving with that kind of active propulsive force towards the front of these groups. The black cells are just moving passively according to the drag force and repulsive forces here. But we can actually get some kinds of overtaking behavior to occur. Again, this is just an early version of the mathematical model. It's probably going to change a lot more in the foreseeable months, but we can at least get this nice thing to occur. The important thing is, how does this affect the statistics that we can collect, such as where these cells are over time, the area that they cover, how aligned the cells are to each other, as you vary the strength of different parameters, such as the number of agents that you have. And then finally, the last project that I'll discuss very briefly is this project line migration, which is a little bit out of my field, but this originally arose through one of the dinners at the Newton workshop, where I was sitting next to Genevieve Finnerty, who's over at the Max Planck Institute for Collective Behavior, and she had this problem about lions migrating, where a lion pride disassociates, and so that like you only have one line moving off on its own, and it goes very far away from the original pride on the order of tens of kilometers away, but is able to somehow manage to, after a period of say like weeks or months, able to reform the pride at a different location from where it started. And I guess the question that Genevieve had, that she thought like Philip and I could answer with a mathematical model, was just, is it just through communication that these lines could actually find their way, or do they need something more complicated like an internal memory? And so the idea is that you would have some kind of signals in your environment. So the lions have some, like you can see another lion at a certain distance. You can hear them, or you can sense some kind of olfactory trail they leave behind. But these factors are all modulated by how long how long it took from when the signal was emitted, how far away you are from the signal, and how well you can actually perceive it. And again, this is in animal migration, which is a little bit outside of my expertise, but does relate a little bit to cell migration, because even in cell migration, you have this problem of signaling across multiple scales where cells can signal each other across a large variety of spatial and temporal scales, and cells might react to it either instantaneously, or it might take a long time to actually respond to that signal. So the idea is we want to understand how the effect of different signals over different scales can affect your responses. And again, still a work in progress here, but the idea that we're having at the moment is to have essentially a run and tumble model where individuals move at a fixed speed, but update their angle of travel depending on how well they perceive a target. And we want to try to assess how well you can actually find the source of that signal just by assessing either a hitting time distribution. We also want to look at how much area this moving individual covers before it's eventually it finds its target. But with that, I think I'm essentially out of time here. Just wanted to leave here with the future plans again. So in terms of the zebrafish patterning, we're looking more at quantifying all these statistics, making sure they're robust to different initial conditions. In terms of blood vessel development and line migration projects, trying to actually finalize these theoretical models and performing some sensitivity analyses to understand how different parameters are affecting how well you can either overtake your neighbors or find a source of a signal. And I would also just like to thank the Isaac Newton Institute and Simons Foundation for funding my stay here, and my collaborators, the ones who are part of the Mathematics of Movement workshop, are highlighted in the box there. And then we also have a paper related, or preprint, sorry, related to the zebrafish project, which came out a couple months ago while I was on the program. With that, thank you much. Have you any questions?
So any questions?
So for the cells, since I looked at the movie of the cells overtaking,
yeah,
in your model, so you put a soft repulsion interaction, or so on the data. So this is 3D and probably somehow. And do they actually not go over? They kind of go sideways.
Yeah. So going over in 3D is a possibility. The experiments that I was showing you, though, were on a glass slide, so it's more of a 2D scenario. Yeah.
So,
in principle, in real life, it's in principle in 3D as well.
So,
yeah.
So the repulsion, it's
not
needed to something. No? They can go over it.
Yeah. Yeah.
Yeah, I was just interested in the kind of assumptions you have that can cause overtaking in these kind of populations. Thank you for the great talk.
The assumptions that we have that are underlying overtaking. Yeah. So we're assuming that there's, each cell has a kind of internal chemical. You can think of it like energy, or it could be some other kind of protein. But you have a single chemical species that each cell has. If you're above a certain level, you are able to propel yourself forward through some kind of like molecular motor, say. Essentially that's the assumptions that we're making for that particular model there. So I mean, in terms of like the angiogenesis stuff, it's a bit more clear cut that the chemicals seem to be delta notch signaling. So it's, in that case, it's 2 signals. We're just trying to think of what's the simplest model that we can actually get to have this overtaking behavior. So it's a bit more abstract in that kind of view.
Thank you, Duncan. Regarding also the angiogenesis model, I was under the impression that you never needed inertia for this kind of movement, but you used
yeah. So there's
really need that.
Yeah, not really. There's a version of this model that we played with where the friction term depended on space, and in that case, you would have to go with the second order model, because if friction becomes equal to 0, then the assumption that you're making about like the overdamped like limit assumption that you're making is a bit more tricky to work with. But yeah, in principle, that version of the model I showed you, we also have a first order version of it, and it's essentially the same. Yeah, so it works better with a constant like drag force term. That makes sense.
Thank you.
Okay.